am Sebastian. This is the third part of the series of videos where we have been discussing about translocational motors and as well as how proteins which are synthesized in the cytoplasm is transported to, for example, into a mitochondria and how it is getting incorporated. And in this video, we will look specifically at the last part, that is incorporation or integration of a protein molecule in the mitochondrial membrane or in the space. There are several mechanisms proposed. We will look at 5 ways by which protein can be integrated. So this is the outer mitochondrial membrane and this is the inner mitochondrial membrane. The protein that we bring inside must get incorporated in the inner mitochondrial membrane, and here we have the Tom complex and here we have the Tim complex. So if you look at the protein, here is the signal will be here, the signal is there, then you have the protein, protein is being pulled inside. Now this protein which has to be incorporated in the inner membrane, it will have one more signal. That is, so if this is the protein to be incorporated and this is the signal, it has an additional signal. That additional signal is, it is hydrophobic in nature. And that signal says where it has to be incorporated. So what is likely to happen is, this red signal is normally removed by a mitochondrial signal peptidase. A mitochondrial signal peptidase will remove the red signal. Once it removes the red signal, so now you have the green signal, that is the signal for integration, where it has to be incorporated. So what is likely to happen is, now this remains here, this is the Tim complex. It will try to pull the protein inside, and after some time the Tim complex move away and this protein gets incorporated in the membrane. So this is how one way of integrating protein in the inner mitochondrial membrane. A second way to integrate protein in the membrane is, assume this is the inner mitochondrial membrane. Now the protein is brought inside. So the protein is, this is the Tim complex and the protein is fully inside now, and this is the signal sequence. Now if the signal is cut and removed by the peptidase enzyme, the signal peptidase, now this is the protein that has to be integrated in the membrane. In the inner mitochondrial membrane, there is another protein complex that is known as OXA. OXA is a protein, and this protein now will take up this protein which has been brought inside. OXA takes up this black protein, takes it up, and it tries to assist the black protein to get incorporated in the membrane, get folded and gets incorporated. So a second mechanism, that is OXA assisted mechanism of incorporating a protein in the membrane. In the exact mechanism, what is the role played by OXA in integrating is not really known. A lot of research work has to be done in order to prove the role of OXA, but it is associated, OXA has a role. This is what has been proved. A third mechanism is, assume this is the inner mitochondrial membrane and here is the protein and this is the signal, that is the signal which got incorporated. What is likely to happen is, if the protein has to be located in the inner membrane space, this portion is cleaved by a peptidase enzyme. Peptidase will cleave, so therefore what will happen? This protein gets incorporated in the inner membrane space. So another way to translocate a protein. So this is the third mechanism. Okay. A fourth way to bring in protein and integrating them is these important proteins, especially in the outer mitochondrial membrane. So we have the Tom complex here. This is bringing in the protein. Now this protein which is brought inside, it has an inner signal, and this inner signal will direct that this protein is meant for a particular location. Now what happens when this protein is in the inner mitochondrial membrane space? So this is the inner membrane. What will happen is there are molecular chaperones present in the inner mitochondrial membrane space. These molecular chaperones will bind on this protein. After binding, these molecular chaperones will help the protein to fold. And in the inner membrane, you have the Tim complex, especially Tim22, will take up this protein along with the chaperones and helps the protein to fold and gets incorporated into the membrane. And so this will be the signal, and the signal which is present in between the protein will help the protein to be incorporated into the membrane. This is the fourth mechanism of integrating protein into a membrane. In the fifth and the final mechanism proposed is by using another protein known as the MIA40 proteins. MIA stands for mitochondrial inner membrane space assembly. Mitochondrial inner membrane space assembly. How is MIA40 helping? So these are especially for soluble proteins which are present in the inner mitochondrial membrane. Now these are proteins which are in the reduced form. So therefore this is SH, it is in the reduced form. And now these proteins which is in the exterior side, that is cytoplasmic side, they are taken inside through the Tom complex. Now once it is inside, this is the outer membrane, this protein is inside. Now this is SH, this is SH. Now the MIA protein will come into picture. What the MIA protein does is, assume this is the MIA40, this is the MIA protein 40. MIA protein exists in the oxidized form. So therefore it is yes, it remains in oxidized form. As soon as this protein comes, it forms a kind of this is a disulfide bond. It oxidizes the protein that is brought inside. It oxidizes. Once it is oxidizing, now the MIA protein is now undergoing from oxidized to, it will undergo a reduced form. In the process of MIA protein undergoing from oxidized to a reduced form, it becomes SH and SH. It will displace the protein, and this protein now will become an oxidized protein with a disulfide bond. So this is the protein that we have brought from outside to inside, translocated using the MIA protein, and this has folded. The protein has been folded. Now it can be incorporated into the membrane. What happens to the MIA protein? It got reduced. Now this reduced MIA protein will encounter the components of the electron transport chain, and it will be converted back into the oxidized form by using the iron sulfur protein, other components of the electron transport chain. Very interesting to observe how the reducing potential is utilized in order to bring a protein, fold a protein, and integrating into the membrane. So these are the 5 ways by which integrating protein, from the cytoplasmic side, bringing them inside, then integrating them into the inner membrane space or in the inner membrane. So we have looked at elaborately on different mechanisms.